So we're going to be progressing from the last one, from the last video, and we stopped at iron storage, right? Iron storage. When we talk about iron storage, there are certain things that we need to know how liver is involved in storing iron. Basically, the RBCs, red blood cells, when they become old, they are destroyed in spleen. Spleen is a small gland. Spleen is a small gland right beside liver. It's present. This gland is actually present right beside liver. So beside liver. All right. So the spleen is going to break down red blood cell. Once the red blood cell is broken down, uh, hemoglobin is produced. Hemoglobin is released. This hemoglobin is going to be, this hemoglobin is brought to liver. All right, it is brought to liver, and this hemoglobin is further broken down into two components in liver. This iron is removed and stored in liver. Secondly, bile pigments are also produced. Bile pigments are also produced. All right. This is what is actually taking place. This is how this iron that is there is stored in the liver. This is how iron is stored in the liver, right? Are you writing this down?
Last point, hemoglobin.
Right. The next is protein synthesis. Talk about protein synthesis. The liver is involved in synthesizing some of the proteins found in the blood plasma. The liver. Adia, can you mute? The liver synthesizes proteins found in blood plasma. All right. Some of the amino acids that are found in amino acids found in diet, right? The non-essential ones. Essential ones are the ones which body cannot make, and they need to be taken through the diet. All right. So some of these proteins that we are talking about include prothrombin. Prothrombin and fibrinogen. All right, and we discussed about it. I think we'll see that they are involved in blood clotting, clotting of blood, right? The next one is an important one, deamination of amino acids, a very important part. Deamination of amino acids. All right. So deamination of amino acids, right? Deamination of amino acids. When you talk about deamination, excess amino acids are actually taken to the liver, and excess amino acids, they are going to be deamine. What the definition that we need to, so what is going to happen? Excess amino acids, excess amino acids are transported to liver, where their amino group is removed and converted and converted to urea. Urea converted to urea. This is called deamination. Okay, this is called
जी
deamination. This is called deamination. So what is going to happen actually, if you look at it, we were able to see this was the structure of amino acid. Remember, there was an R group and there was this R group is like a fingerprint of the amino acids. It's like a fingerprint of amino acids. Uh, each amino acid is distinguished by this R group. All right, it could be there are many different, and that makes different around 21 amino acids. So what happens is that this amino group is removed. It is removed. It is dropped. It is removed, and it is convert. And once it is removed, it is converted into ammonia. All right, but this ammonia is toxic to cells. Toxic to cells. It is toxic to cells. So it is readily converted into urea. And now urea is a non-toxic nitrogenous waste. Nitrogenous waste product that is removed. This is removed in urine. All right, removed in urine. You understand? So you're going to be left with this part. This part is actually taken as it is converted into a carbon residue. Carbon residues we are going to study. They are used in many other processes, making many other organic compounds in the body, particularly carbohydrates. So it is converted into glucose, and this glucose is then converted into glycogen for storage. Glycogen for storage in liver. All right, is this clear? Stored in liver. What we need to understand over here, glycogen, it doesn't mean that it is only the liver in the body that converts glucose into glycogen. There are many other, this is also done in the muscles. Muscles also respond to insulin. All right, it is not only liver that responds to insulin, muscles also respond to insulin, and they convert excess glucose into glycogen. And they can also respond to glycogen, and they can convert it into they can also respond to glucagon and convert glycogen into glucose. Do you understand? Plus, the muscles
जी
and liver, they also respond to adrenaline, and they respond to that, and in the presence of adrenaline they are also converted into the glycogen is also converted into glucose. All right, clear? Any questions regarding this? Are you writing this down?
जी ऊपर कर दें थोड़ा सा
Hmm.
डेमाइनेशन से डीमाइनेशन ऑफ अमाइनो एसिड्स
Deamination of amino acids.
और सर आपका कैमरा भी खुला हुआ है
सही है. Last one. The last one is the detoxification. Detoxification by liver. This is one of the most, you know, a very important function of liver that takes place by liver. Detoxification process, in which what is the process? Basically, the process of converting toxic substances into harmless ones is called detoxification. All right, detoxification. As we saw, the urea, one of the examples is ammonia being converted into urea. All right, from a harmful to a non-harmful. All right, so one the important thing that basically the syllabus outlines for us to understand over here is the is alcohol is actually liver actually detoxifies alcohol into useful substances. So what when the alcohol is consumed, what does it do? The liver uh, breaks breaks down alcohol into acetyl acetaldehyde. Acetaldehyde. Now, this is done by liver cells have enzyme called alcohol dehydrogenase. Alcohol dehydro alcohol dehydrogenase. All right. This is the adaptation of the liver cells. They have this enzyme alcohol dehydrogenase which converts alcohol into acetaldehyde, and this acetaldehyde can be further it is further broken down to make useful products like products such as respiratory substrates. Okay, you don't need to go into that detail, but there is obviously this is this toxification process takes a lot of energy, but excessive consumption of alcohol also has a bad effect on the liver. All right, so uh, if the as if you now by looking at the functions of the liver, we understand that if the liver is affected, if liver starts to become disease, if alcohol abuse basically causes liver. Long term alcohol abuse, consumption of a lot of alcohol leads to, causes liver to cause causes liver to have cirrhosis. It's called liver cirrhosis, a disease in which the liver cells are destroyed. A disease in which liver cells are destroyed and replaced with fibrous tissues. Cells are destroyed and replaced by fibrous tissues. Hardening of liver, damage, a lot of damage that might lead to other, uh, it leads to carcinomas of liver as well, and it leads to, the disappearance of the function of liver as well, replaced by fibrous fibrous tissues. Now this makes the liver un- less able to function properly. All right, liver becomes less functional. Liver becomes less functional. All right, and because of a lot of damages to the liver, the, the the whole anatomy of liver, what happens is that sometimes there is a chance of internal bleeding of liver as well. Hemorrhage, we can also observe hemorrhage of liver. All right, internal bleeding of liver. This leads to liver failure and subsequently death. All right, this is what, you know we actually had to do in the topic of digestion, which is human nutrition. All right, and we are already done with the nutrient topic as well, right, Nadia? What we are supposed to be doing from here onwards, we are going to
Mm.
go through because there are going to be a lot of questions in your exam. Obviously, from this topic, it has a very good weightage because these are two topics combined together actually. So, inshallah, we'll go through the past paper for this. Inshallah, I want you to write this down. Inshallah, then we'll move forward.